known living things, you must have wondered and asked yourself, what is it that makes an organism living? Or what is it that an inanimate thing does not have, which a living thing has? The answer to this is the presence of the basic unit of life, the cell in all living organisms. All organisms are composed of cells. Some are composed of single cells and are called unicellular organisms, while others, like us, composed of many cells, are called multicellular organisms. What is a cell? Unicellular organisms are capable of independent existence and performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural detail of the cell. Cell theory. In 1838, Matthias Schleiden, a German botanist, examined a large number of plants and observed that all plants are composed of different kind of cells which form the tissue of the plant. At about the same time, Theodore Swan in 1839, a British zoologist, studied different type of animal cells and reported that cell had a thin outer layer. They reported that cell had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plants' tissues, that the presence of cell wall is a unique character of the plant cells. On the basis of this, Swan proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. Schleiden and Swan together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow in 1855 first explained that cells divided and new cells are formed from pre-existing cells. Omnis cellula e cellula. He modified the hypothesis of Schleiden and Swan to give the cell theory a final shape. Cell theory, as understood today, is first, all living organisms are composed of cells and products of cells. And the second is, all cells arise from pre existing cells. An overview of cell. You have earlier observed cells in an onion peel and or in a human chick cell under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundaries, and just within it is the cell membrane. The cells of the human chick have an outer membrane as a delimiting structure of a cell. Inside each cell, the cell of human chick have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane bound structure called nucleus. This nucleus contains the chromosomes, which in turn contain the genetic material, DNA. Cells that have membrane bound nuclei are called eukaryotic, where cells that lack of membrane bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cells, a semi fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both plants and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane bound distinct structure called organelle, like the endoplasmic reticulum ER, the Golgi complex, lysosomes, mitochondria, microbodies, and vacuole. The prokaryotic cells lack such membrane bound organelles. Ribosomes are non membrane bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the tuned organelles, chloroplast, which is in plants, and mitochondria, and on rough ER, endoplasmic reticulum. Animal cells contain another non membrane bound organelle called centrosome, which helps in cell division. Cells differ greatly in size, shape, and activities. For example, mycoplasmas, the smallest cells, are only zero point 0.3 micrometer in length, while bacteria could be in 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich among multicellular organisms. Human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. Now, prokaryotic cell. PPLO is a type of mycoplasma. PPLO full form is pleuropneumonia like organisms. Red blood cells are round and biconcave or disc shaped. White blood cells are amoeboid in structure. Columnar epithelial cells are long and narrow. Nerve cells are branched and long. A tracheid, which is elongated cell structure. Mesophyll cells, round and oval. Prokaryotic cells. In prokaryotic cells, the prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, which is rod-like, coccus, which is spherical, vibrio, which is coma-shaped, and the spirillum is spiral-shaped. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane except the mycoplasma. The fluid matrix filling the cell is the cytoplasm. There is no well defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA is the circular single chromosome or circular DNA. Many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. The plasmid DNA confers certain unique phenotypic character to such bacteria. One such character is resistance to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelle like the one in eukaryotes are found in prokaryotic cells except ribosomes. Ribosomes are found in prokaryotic as well as eukaryotic cells. Prokaryotes have something unique in the form of inclusion bodies. A specialized differentiated form of cell membrane called mesosome is the characteristics of prokaryotes. Mesosome are the infoldings of plasma membrane in the prokaryotic cells. They are essentially infoldings of cell membrane. Now, cell envelope and its modifications. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure. Example is the outermost is glycocalyx, followed by the cell wall, and then the plasma membrane. Although each layer of envelope performs distinct function, they act together as a single protective unit. Bacteria can be classified into two groups of the basis of the differences in the cell envelopes and the manner in which they respond to the staining procedures developed by Gram-Wiss. Those that take up the gram stain are called gram positive bacteria, and the others that do not are called gram negative bacteria. Glycocalyx differs in compositions and thickness among different bacteria. It could be a loose sheath called the slime layer in some, while in others it may be thick or and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting and collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is The mesosome, which is formed by the extension of plasma membrane into the cell. These extensions are in the form of vesicle, tubule, or lamellae. They help in cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration, secretion process, and they also help in respiration, secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes, like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which contain pigments. Bacterial cells may be motile or non motile. If motile, they have thin filamentous extension from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, pili or fimbriae are also surface structure of the bacteria but do not play a role in motility. The pili are elongated tubular structures made of a special protein, pilin protein. The fimbriae are small bristle-like fibers sprouting out the cell. In some bacteria, they are known to help attach the bacteria to rock in steams and also to the host tissues. A ribosome and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer to 20 nanometer in size and are made of two subunits, 50s and 30s units, which even present together form 70s prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysome. The ribosomes of a polysome translate mRNA into proteins. Now, inclusion bodies. A reverse material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. Example: phosphate granules, synophysin granules, and glycogen granules. Gas vacuole are found in blue-green algae and purple and green photosynthetic bacteria. Now we move to the eukaryotic cells. The eukaryotes include all the protists, plants, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possess cell walls, plastids, and a large central vacuole which are absent in animal cells. On the other hand, animal cells have centrioles which are absent in almost all plant cells. Let us now look at individual cell organelle to understand their structure and functions. Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells, which is called RBCs, also called ghost cells, enabled the scientists to deduce the possible structure of plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tail toward the inner part. This ensures that the non-polar tail of saturated hydrocarbons, which is a single bond present in the hydrocarbons, is protected from the aqueous environment. In addition to phospholipid membrane, also contain cholesterol. Later biochemical investigation clearly revealed that the cell membranes also possess protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocytes has approximately 52% protein and 40% of lipids. Depending on the ease of extraction, membrane proteins can be classified as integral protein, peripheral protein, integral which is uh, completely embedded in the membrane, peripheral which is present in the outer side to the membrane. Peripheral proteins lie on the surface of the membrane, while the integral protein are partially or totally buried in the membrane. So these are the structure of fluid mosaic model of plasma membrane. Here are we can see the polar head and hydrophobic tails, integral proteins, phospholipids bilayer. This is glycoprotein, this is glycolipid. These are only sugar which is only present at the outer side, and this is the peripheral protein which is only present in the outer side of the membrane, and there are cholesterol present in between of the cell membrane at certain distance, continuously at the certain distance. An improved model of the structure of cell membrane was proposed by Singer and Nicholson. Singer and Nicholson. Singer नहीं है Singer. Singer and Nicholson in 1972. Widely accepted as fluid mosaic model. According to this model, the quasi-fluid nature of lipid enables lateral movement of protein within the overall bilayer. This ability